In this video, we will be exploring the significance of target-mediated drug disposition, or TMDD, in hemophilia treatments. Overcoming TMDD by modifying a drug's binding affinity can have valuable implications for patients who currently depend on treatments with frequent dosing. What is TMDD? TMDD, or target-mediated drug disposition, is the process in which a drug binds with high affinity to its target to such an extent that it affects the drug's pharmacokinetics. This involves how a drug is absorbed, distributed, metabolized, and eliminated following administration. While TMDD pharmacokinetics apply to a number of common therapeutics, it occurs primarily with large molecule drugs, such as monoclonal antibodies. Some common features of large molecule drugs that undergo TMDD include high target binding affinity, high target specificity, limited receptor capacity for ligand binding, and rapid clearance at low dose ranges. A drug undergoing TMDD exhibits a nonlinear, dose-dependent pharmacokinetic profile, which means that the concentration of the drug in the body rises disproportionately with an increase in dose. TMDD nonlinearity is often seen at intermediate to lower drug concentrations, following a single-dose regimen, with a rapid and significant decrease in systemic drug concentration due to target binding and saturation. A typical TMDD curve begins with A. An initial rapid drop in drug concentration caused by the fast association of the drug and target. B. The target then becomes saturated, and drug elimination occurs mainly by a first-order process where the rate of elimination is directly proportional to the drug concentration. C. This is followed by a nonlinear transition phase with mixed elimination of the free drug and movement of the drug-target complex into the cell. D. When most of the free drug has been eliminated, another linear decline phase occurs due to the slow decrease of drug concentration. Why is TMDD important? The influence of TMDD on drug elimination profiles is important to understand, since pharmacokinetics in the nonlinear range may lead to possible variability in efficacy and safety. How can TMDD be overcome? Modifications can be made to overcome TMDD and to ensure a more predictable relationship characterized by increased linearity between drug exposure and response. The dosing regimen of a drug can be optimized, with higher doses given to saturate the target, or more frequent doses given to maintain target concentrations. Administering high or frequent doses can help to maintain the therapeutic concentration of active drug in the body. The binding affinity of the drug may also be modified. Important differences in binding affinity of monoclonal antibodies can affect the dose and frequency of administration needed to maintain target concentrations. Large molecule drugs with a higher binding affinity may require higher doses and/or more frequent dosing in order to offset high clearance rates and restore linear clearance. A drug that has been modified to have a lower affinity may exhibit slower clearance rates and may offer less frequent, fixed dosing for patients. Healthcare providers may be accustomed to managing dosing with conventional treatments for hemophilia, such as factor VIII or IX replacement therapies. Understanding TMDD could help explain why some large molecule drugs are more effective at certain doses or within specific time intervals. Factor replacement therapies and certain monoclonal antibodies are rapidly eliminated and therefore require more frequent dosing to maintain large concentrations. mAbs with higher affinity binding exhibit a more pronounced TMDD and show a steep decline. mAbs with a lower binding affinity show a lesser degree of TMDD due to drug modifications made to overcome TMDD, therefore exhibiting a slower decline. The ability of a drug to overcome TMDD and restore linear clearance at clinical doses while maintaining a favorable risk-benefit profile may have valuable implications for patients who currently depend on frequent infusions. One approach to overcoming TMDD is to design the drug with lower binding affinity. This may enable less frequent, fixed-dose subcutaneous administration without the need for weight-based adjustments. For further information on TMDD, please contact your Pfizer medical team.